Okay. So chapter 20 again is another. I say it's a fun chapter, and it is. So chapter 20. Here we go. So first slide, right out of the gate, you're going to see a term there that you probably haven't seen in a little bit, but we've discussed it, and this is why it's very important, and I stress it quite a bit in my other 210 classes. Is we're going to be talking about movement of substances, especially when we get to the circulatory system where we are now, and so we need to review these concepts. So diffusion and vesicular transport. Diffusion, okay, remember it's a passive process, which means it doesn't require any energy, ATP, and you're going to move a substance, a solute, from an area of high concentration to low concentration. So when we're talking about the circulatory system and we're seeing how things enter the bloodstream or leave the bloodstream, right? Diffusion plays a large role in that. And it plays a large role because of our concentration gradient that has been established. Right? So when you look at the stuff that is flowing in your blood, okay, you're going to have all sorts of stuff besides your foreign blood elements. All right, in the plasma, you're going to have lots of proteins. You're going to have hormones, nitrogenous wastes, metabolites, nutrients, or respiratory gases. So things like oxygen and the hormones there and the nutrients, all the stuff that needs to maybe go to a target cell or our cells, they're going to move from not to, but from the blood to the interstitial fluid. What is that? Well, the interstitial fluid is that fluid that's right outside of the blood vessels. And then conversely, carbon dioxide and wastes are going to move, all right, from the cells, and then that stuff will leave the cells and it'll go into the interstitial fluid, and then from the interstitial fluid, that material will diffuse, all right, from the tissue to the blood. So the good stuff leaves the blood and goes into the interstitial fluid, and then the bad stuff leaves the interstitial fluid and goes into the blood. Right. So again, a lot of this is based on the concentration gradient. Also, though, right, when we talk about how or what route of diffusion, right, is going to be utilized, it plays the size of whatever it is that we're discussing, the particle size, plays a large role. So obviously, and again, you learned this before, with oxygen and carbon dioxide, respiratory gases, they can diffuse directly through the plasma membrane. They're very small, they're nonpolar, okay? So they slide right through, right? And so that's what we're going to see. So oxygen can slide in between the cells or right through the plasma membrane. Larger solutes, all right, for example, proteins come in various sizes, right? It's a little bit harder for them to leave the bloodstream, right, in the capillaries, unless we make certain accommodations for them. And we talked about this in lab class. Our capillaries come in 3 different types: continuous, right, fenestrated, and sinusoidal. And it's the fenestrated and sinusoidal that will have these holes in the actual cells, right, in the endothelium there. And so larger solutes can then pass through those holes. The sinusoids are much larger than the fenestrations there, right? That makes a big difference there. That's the plan. I knew it. Knew it. Okay. So when we're discussing how things move, right, from point A to point B, either from the blood to the tissues and vice versa, right? Diffusion is one way. Vesicular transport is another. Now, this goes back to chapter 4 when you were learning about active, passive transport processes. And this goes back to penocytosis and exocytosis. Penocytosis is that we nicknamed it the cell drinking, where substances that are outside, they're usually fluid type substances that are outside the cell, and the cell just kind of extends these little kind of pseudopods around, right, those substances, and then forms a bubble around it, okay, a vesicle, and then it'll pull it to the inside. Exocytosis, think exit, right? We are going to get rid of stuff that's inside the cell and push it to the outside of the cell. So essentially, right, when we're talking about vesicular transport, we're talking about how we can move things right, through a cell. So pinocytosis, we take it in. Exocytosis, we get rid of it. Keep in mind, this goes in both directions. So larger substances that can't directly diffuse, right, through the plasma membrane or have a hard time getting into the cell, we can use vesicular transport to facilitate this, right? Fatty acids, hormones, several other substances, right, that can be utilized for this, right, when we're trying to move material through the cell inside and out. Now, this brings me to bulk flow. It's an amazing concept. I know I say this a lot, right? But bulk flow is new to us, but pressure gradients are not. So the definition of bulk flow is fluids flow down a pressure gradient. Well, gosh darn it, we've seen that before. We saw it in the heart, right? And so we saw that blood moves from an area of high pressure to low pressure. Same situation here, okay? So fluids, right, and usually we have dissolved substances in those fluids are going to move, right, from an area of high pressure to low pressure. Here's the thing. When we're dealing with our blood vessels, it's not going to be just one, all right, pressure, all right, system working on the fluid. We're going to see here in a moment that you have several different types of pressures that are going to be directed on the fluid, and therefore, right, it's like a tug of war, right? You got several people on a team, and the teams are opposing one another, and they're tugging on that rope, all right? Okay, so you have multiple factors involved, right? But eventually, you're going to see that rope move one direction or the other. Same thing here with bulk flow, right? We're going to see what the net pressure is, and they're going to be opposing forces. So some pressures are pushing in towards the blood vessel, and some pressures are pushing out from the blood vessel out into the tissue there. So this brings us into what we call the when we're talking about bulk flow, we're going to be discussing, right, these two pressures here, hydrostatic pressure and colloid pressure. And we've talked about both of them just in passing in previous chapters here. And we'll go back and we'll hit them up in a second here. But the two terms listed here, I want to discuss real fast: filtration and resorption, right? When you're thinking of filtration, you want to think of fluid that's moving out of the blood. So think about the water pipes in your house or wherever you are, right? And as water is flowing through those pipes, it's pushing on the walls of the pipes. And so essentially it's trying to push out and get out of the pipe, right? So when we're talking about filtration, that fluid is going to try to move out of your blood, right? So fluid and very small solutes can move through some of those openings in the capillaries, especially if they're fenestrated or if they're sinusoidal. A little bit harder if it's continuous, can still be done, right? But regardless, that can still happen. So the nice thing about it is the large solutes are blocked. Red blood cells, white blood cells, platelets, all right, large proteins, even some intermediate or medium-sized proteins, those are blocked. Right, so when we're talking about filtration, we talked about this a little bit in lab class when we were talking about chapter 20. We're going to see filtration mainly at the arterial end of the capillary. Remember that picture? You had the capillary bed, and you're looking at the picture. The left side, you had the arterial. On the right side, you had the venule. So on the left side of that capillary bed, you're going to see fluid that's going to try to move out of the capillaries into the tissue, which is good, especially when we're trying to offload nutrients and oxygen and hormones and all the other stuff that the cells could use. Now, resorption, right? I want you to think fluid moving back into the blood. So it's in the interstitial fluid, and it's going to try to get into the bloodstream. So this is going to be seen at the venous end of the capillary bed there that we saw in lab. Right, that'll be on the right side if we were looking at that picture. And I think I got a picture here somewhere. There you go. You can kind of see it here. So on this side here, on the left side, this is the arterial. Here's the venule. So fluid is going to try to leave the capillary here, and then it's going to come back over here. Come back inside of into into the capillary there. Okay, so let's do some defining here, and let's talk about hydrostatic pressure. Okay, again, it's defined as the force exerted by a fluid, and usually, like I said, you want to use the analogy with water in your pipes. It's that force that is behind the water trying to, if you have a leak in the pipe, it's going to leave the pipe and go into your kitchen floor. So we have two types of hydrostatic pressure. The first one here is the blood hydrostatic pressure. Okay, that is the pressure that your blood exerts on the vessel wall as it's flowing through your blood vessels there. So when we get to the capillary, like I said, right, fluid is going to leave and go into the capillary bed on the arterial end or the artery end, and so that's going to be filtration. It's going to leave the blood and go into the fluid. And then the other type of hydrostatic pressure is what we call interstitial fluid hydrostatic pressure. And so this is the force that the interstitial fluid that's outside of the blood vessel is exerting to go back into the blood vessel. So it's pushing on the outside wall, pushing, and it's trying to get in. Keep in mind, in most of our tissues, this is 0. You just memorize that. All right, so this fluid is, this pressure for the interstitial fluid is going to be 0. All right. The other type of pressure, and we've discussed this before, right? When we were talking in chapter 18, when we were talking about blood, the colloid osmotic pressure, consider what those terms mean. Colloid, think protein. Osmotic, think water. All right. So it is the pull on water due to the presence of proteins, our colloid. So this is that kind of like attractive force. So we have two types of colloid osmotic pressure. The blood colloid osmotic pressure. Again, we have all those proteins in your blood, right? The big main class are the albumins that are made in the liver. And so they're going to try to draw water into the blood vessel, into the capillary. All right, and or whatever water that's inside the capillary, it's going to try to keep it in the capillary and prevent it from leaking out. All right, so here, when we're talking about blood colloid osmotic pressure, it's mainly going to be involved in resorption. So this blood colloid osmotic pressure is in opposition to your hydrostatic or your blood hydrostatic pressure. So the blood hydrostatic pressure is trying to push on the walls of the blood vessel and try to leave the capillary, and that's going outwards. Meanwhile, you got your blood colloid osmotic pressure, which is going to be pulling. We're trying to keep that water in the blood vessel here. Just to let you know, when I was learning all this material, we did refer to it as oncotic pressure, but we're seeing it referred to more as colloid osmotic pressure now. All right, the other type of colloid osmotic pressure is the interstitial fluid colloid osmotic pressure. So again, now we're outside the blood vessel in the interstitial fluid. And so we're going to see here, because one of the things, if you don't remember, in chapter 18, we said that the composition of blood, the fluid, blood inside of a blood vessel, right, when it comes to the plasma, is very similar to the composition of the fluid in the tissues outside of your blood vessels, except for one very big thing. There's hardly any proteins in the interstitial fluid, right? All the proteins, or most of those proteins, will be in the blood vessel, in the capillary there. So that's going to be the big difference there. So in the interstitial fluid colloid osmotic pressure, right? Like I said, this pressure draws that fluid into the interstitial fluid because it's got like an attractive force there. But since we have so few proteins in the interstitial fluid, it's going to be a relatively weak attractive force. So 0 to 5 millimeters of mercury of pressure being exerted there. Right? So, okay. So think about it. Interstitial fluid hydrostatic pressure, close to 0. And interstitial fluid colloid osmotic pressure, anywhere from 0 to 5 milligrams of mercury. So there shouldn't be too much force, all right, out in the interstitial fluid, all right, when we're talking about hydrostatic pressure and colloid osmotic pressure. It seems like most of the force or pressures and whatnot are going to be strictly kind of relegated to the inside of the blood vessel, as it should be. Okay, so we just learned about the different types of pressures. Okay, we learned about hydrostatic and then the colloid osmotic pressure. So now what we're going to do is talk about, and I'll wrap it up with this, for net filtration pressure. So what we're going to do is we're going to add all these pressures up because we're trying to determine, right, when it's all said and done, is the force inside the capillary enough to have stuff leave the capillary, or is the force, all right, inside the capillary not enough for stuff to leave the capillary, but stuff will enter into the capillary from the tissue side? And that's what we're going to see here. Right, so that's essentially what net filtration pressure is. It is going to be, we're going to take both hydrostatic pressures and take the difference from those. And then we're going to take both of the colloid osmotic pressures and then take the difference of those. And then we're going to essentially subtract or take the difference of the colloid osmotic pressure away from the net hydrostatic pressure. So what does that all mean? Look at this equation here. This is one of the equations that you have to know, all right, for your class. So net filtration pressure is the difference between your blood hydrostatic pressure and then the interstitial fluid hydrostatic pressure. This value is usually going to be greater than this value here, at least by the arterial end. So you're going to subtract blood hydrostatic pressure, all right, or excuse me, you're going to subtract the interstitial fluid hydrostatic pressure from the blood hydrostatic pressure. And then we're going to go over here to the colloid osmotic pressures. You're going to subtract the interstitial fluid colloid osmotic pressure from the colloid osmotic pressure in the blood. And then once you get those two values, then you're going to subtract your colloid osmotic pressure from your hydrostatic pressure. All right, and that's basically what we're seeing here. So the net hydrostatic pressure is the difference between blood and interstitial fluid hydrostatic pressures. The net colloid osmotic pressure is the difference between the blood and the interstitial fluid osmotic pressures. All right, and so on the arterial end, like I said, the net filtration pressure should favor filtration, which means material is going to move, all right, fluid's going to move from inside your capillary out into the tissues. And then the venous end, all right, the net filtration pressure is going to favor resorption, which means fluid that's in the interstitial fluid, right, outside of the capillaries is going to enter into the capillary. So that's what this picture here is showing us. So I'm going to kind of walk you through it. So here comes the blood from the heart in the arterial. It enters into the capillary here. All right, so we measure here on the arterial end, right, our different pressures. So the first pressure that we're going to measure is going to be our net hydrostatic pressure. So the hydrostatic pressure of blood is 35 millimeters of mercury. And then you heard me say earlier on, the hydrostatic pressure of the interstitial fluid is pretty much going to be 0 in most tissues. So that's where we get this number from. So this number here comes from the hydrostatic pressure of the blood pushing out of the blood vessel. This number here comes from the fluid here that's pushing to get inside of the blood vessel, which is pretty much 0. So we take the difference, which is 35 millimeters of mercury. Right? So there's our net hydrostatic pressure. Now, staying with our equation, we got to figure out the osmotic pressure there, or the colloid osmotic pressure. So we measure and see, okay, that the colloid osmotic pressure in the bloodstream is 26 millimeters of mercury. Why? Tons of proteins here, trying to pull stuff, keep stuff in the capillary, so it's not leaving. Right? And then we'll see, we take the colloid osmotic pressure of the interstitial fluid, which is anywhere from 0 to 5 millimeters of mercury. So we take the difference between those two values, right? 26 minus 5 is 21. So you have 21 here for the net colloid osmotic pressure. You've got 35 millimeters of mercury, which is the net hydrostatic pressure, right? And all we're going to do now is take the difference of these two numbers, which is 14 millimeters of mercury. If it's a positive number, it will favor filtration. Okay, so this positive number here shows that it's favoring filtration, and so fluid will exit out of the capillary. So oxygen is leaving with that fluid. Hormones are leaving. You're going to see glucose, nutrients, all sorts of stuff is leaving the capillary, going out into the interstitial fluid here. All right, now as we move across the capillary and we're getting to the venous end, same type of situation for the net filtration pressure. We're going to use the same equation, but now we're going to see some changes in the pressures. All right, so here again, we're going to then measure, right, the hydrostatic pressure inside the capillary, which is going to be 16 millimeters of mercury. Notice it dropped a little bit. It went from 35 millimeters of mercury to 16 millimeters of mercury because pressure starts to drop as we move from one end of the capillary to the other. So that the hydrostatic pressure is dropped, okay, because we've lost some stuff because it left. That fluid went out. So the pressure starts to drop a little bit. Again, remember, the interstitial fluid hydrostatic pressure should still be 0, okay, because we're not losing proteins or anything, so there's virtually no proteins out here. So that value is 0. So we take the difference, which is 16 millimeters of mercury for the net hydrostatic pressure. And then we get over and start to calculate the net colloid osmotic pressure. So the net colloid osmotic pressure inside of the capillary should not have changed. 26 millimeters of mercury, 26 millimeters of mercury, what we saw in the prior equation, right? Because the protein's composition hasn't changed in your blood, right? And then, again, the fluid interstitial composition of proteins should also not change unless there's some sort of pathology, anywhere from 0 to 5 millimeters of mercury. So that value stays the same. So we take the difference, and we have 21 millimeters of mercury. We take the difference of the net hydrostatic pressure and the net colloid osmotic pressure, and you'll notice we get a negative number here. And so because it's a negative number, it's going to favor resorption. And fluid then is going to enter in to the capillary. And so again, we'll pick up some cellular wastes, we'll get carbon dioxide that enters in, goes into the venous system, goes back to the heart. Oh, perfect. Perfect time to stop